Okay, by now y'all have heard the news about that cool new suspected biosignature they found on Mars, right? Just in case, here's a quick recap. For the last year, scientists at NASA have been analyzing some pretty incredible data generated by the Perseverance rover down in Jezero crater on Mars. And they recently published a bombshell paper in Nature that pretty compellingly describes, well, an analysis of these little spots they found on some rocks on Mars. Now, I'll explain more in a second, but in short, after all the exhaustive analysis we can do from like 100 million miles away, it really does look like the only plausible explanation for these spots is that they were made by living things, like bacteria. And while this paper is very exciting for folks like me, we've honestly had so many quote-unquote evidence of life on Mars moments that have ended up getting debunked or turned out to be inconclusive over the years. And this may very well be one of those. But it's going to take a while to pick apart and verify the findings of this paper, and speculating on whether or not this finding is legit is miles above my pay grade. And so instead of adding to the pile of videos that have rightfully been gushing about this paper, I want to go one layer deeper and answer a question basically no one has addressed in this PR hurricane. What kind of living system leaves behind little rock spots like this? Exploring this question gives us incredible insight into life on the early Earth, how the Earth itself might have shaped the initial spark that produced life in the first place, and what we should be searching for as we try to answer the question of whether or not we're alone in the universe. And to do that explanation, we get to hang out with one of the wildest living things that has ever clung to our planet. That's right, folks, today we're going to meet GEOBACTER SULFURREDUCENS. This near-ubiquitous bacteria gets poisoned by oxygen, so it burrows deep into places like swamps, and breathes by plugging into metal deposits using biological wires. No joke, we're talking about electrobacteria today. It's time to explore metal-reducing respiration and all the incredible hacks bacteria have to do if you can't breathe using oxygen. Let's get into it. And hey, welcome back to Clockwork, a show about the chemistry powering your foundations. Subscribe if you want a better understanding of how your chemistry roots you to the world, and check out our community on Patreon to see our latest sticker packs and papercraft molecules, as we build something really cool over there. But anyway, let's briefly go over the findings of this NASA paper before we get too deep into the biochem. Plus, I really love this space stuff too. Indulge me, I'll make the context fun, I promise. For background, we've got this SUV-sized robot that NASA landed on Mars back in 2021. The Perseverance rover. Old Percy was hurled at this place on Mars, the Jezero crater, as it really looks like there might have been liquid water here once. In the near 5 years since landing, Percy has basically confirmed there used to be liquid water on this part of Mars, but the findings for this paper will be far more significant for the legacy of Perseverance if they end up being true. Let's dig into those. Now on Mars, to the west of the crater floor where Perseverance landed, just past an ancient river delta, there's this little rocky area called Neretva Vallis, where Perseverance took these pictures and did analysis of some wind-worn rock back in 2024. This spot was probably like a muddy river or lake bed before and had some organic matter in the mix. Organic here just means stuff made out of carbon, not necessarily living stuff. And mixed in with that evidence for ancient organic bits were these little spots that all the fuss is about. Zooming in real far to the evidence, you've got a central core of one kind of rock here and darker rock surrounding it. This appears to be something called a reaction front, where there's some sort of chemical change happening on the edges of this spot here. This material in the middle is greigite, and the darker rock is probably vivianite. Vivianite is the key focus for us. It's a mix of iron and phosphate that only forms in really extreme circumstances, or when bacteria are using iron as a material for respiration in the absence of oxygen. Now, most of this paper goes on to discuss all the exhaustive tests and checks the NASA team did to make sure they completely ruled out a non-biological explanation, which is great science, honestly, if you have the stomach for geology papers. You should check that out in the description. There's also a lot of great videos that go into what else this combination of vivianite and greigite could be, and you should definitely go watch those if you haven't already. And I mean, heck, you're probably only here because you did. I don't have to tell you that. But until we can either send geologists to Mars or find a way to make the Mars sample return mission more feasible, there just isn't a lot of conclusive analysis we can add to this paper using this data. It's an exciting result, and it hopefully will add to the motivation we have to go through the ludicrous engineering challenge of bringing some Mars rocks back for human scientists to poke at. But for now, this dataset is really just a compelling question mark, and therefore let's focus on questions we can actually answer, especially since this is a biochemistry channel and not like a space channel. So it's more important for us to pivot and look at what produces vivianite here on Earth. How on earth is vivianite a biosignature? What living process leaves behind an iron mineral crystal? That sounds baffling to me. Vivionite gets produced here on Earth in lots of different ways, but a lot of times it's the leftovers of bacterial metabolism. Specifically, a class of fellas called dissimilatory metal-reducing bacteria, which I know is a mouthful. Basically, metal reducers quote-unquote breathe by grabbing electrons from their food source and shoving them onto metals, specifically a form of iron called iron 3, which, to oversimplify, is a form of iron that's missing 3 of the electrons it would prefer to have. This gives iron 3, or ferric as it's sometimes called, a strong positive charge that can pull on negatively charged electrons really well, which makes it a great candidate for respiration without oxygen. But of course that begs the question, how exactly do you breathe metal? To answer that, let's first meet a metal breather that's really well understood. Now, there are lots of different dissimilatory metal-reducing bacteria that can leave behind greigite and viviite in the combination that Perseverance observed on Mars. Groups like Schwannella immediately come to mind, but we're going to zero in on one of the best researched members of this incredible little group, GEOBACTER SULFURREDUCENS. You're going to find this guy breathing rocks in basically any watery environment that is suitably protected from the pernicious influence of oxygen. We're talking like peat bogs, the deep ocean, certain pockets of the soil, etc. And just on the surface, old GEOBACTER here looks so weird, right? This hairy look the GEOBACTER's got going on is critical to their metabolism, which is the main point of this video. That and the incredible molecular work it takes to respire using rocks. But wait, let's start off with some background real quick. Compared to G. sulfurreducens, your metabolism is honestly easy to understand. Your cells are going to break down your food into fundamental molecules, which your mitochondria are going to, well, burn in order to strip electrons off them. Congratulations on suffering through the world's most oversimplified explanation of the Krebs cycle, by the way. Well, that or the citric acid cycle if you're one of the cool kids. Those electrons are then shoved onto carriers like NADH and FADH2, which are then pulled along the electron transport chain in the inner membrane of your mitochondria. You siphon a little energy from this electron flow to turn the turbine of ATP synthase, thereby powering every single thing your cells do. But to keep this basic electron flow moving, it's going to need a powerful vacuum to pull electrons down the chain. That's why we need the oxygen in the air we breathe. O2 has one of the strongest quote-unquote pulls for electrons in nature. Oxygen is incredibly electronegative, so it provides an incredible sink for electrons to flow into. As long as you're breathing, the electrons in your mitochondrial membrane will flow. And the interesting part here is that oxygen hasn't always been a big part of Earth's atmosphere. O2's electronegativity makes it incredibly reactive, and we have only established and maintained an oxygen-rich atmosphere thanks to the advent of photosynthesis and the constant work of cyanobacteria in the ocean. And like trees too, I guess. Which is one of many reasons why GEOBACTER is an important model organism. Not only are these little critters a critical window for understanding how life could have evolved in the pre-photosynthesis version of Earth, but their metabolism means that they actually end up growing in super weird spots, like literally on electrodes. So there's mountains of research using these guys to develop tech called microbial fuel cells and all sorts of other applications. But I'm going to get into that later because I'm getting distracted. So let's spend a couple of minutes in the deep biochem exploring how GEOBACTER SULFURREDUCENS quote-unquote breathes using metals. Finally getting to the good stuff, GEOBACTER metabolism. And we're going to peel this back layer by layer. At the simplest level, we can chart GEOBACTER's respiration like this. We need a source of electrons to consume, and then a sufficiently attractive place to put those electrons. And so iron 3 is going to be our electron sink, the positively charged vacuum that pulls electrons across this chain. The electron source for GEOBACTER is remarkable. GEOBACTER SULFURREDUCENS is going to use the exact same Krebs or citric acid cycle you do to break electrons off food. But what's the fuel here if you can't use oxygen? Turns out GEOBACTER loves powering its metabolism with vinegar. Straight up acetate ions eventually get processed into acetyl-CoA, and that runs through the Krebs cycle, eventually throwing electrons onto carriers like NADH, NADPH, and even exotic compounds like reduced ferredoxin, which we haven't seen since my photosynthesis series. Nice. And yeah, let's tackle some annoying chemistry lingo before it gets out of hand. I'm throwing the word reduced around on the open internet a little too loosely. Here's the gist. So much in biochem comes down to where electrons are flowing. For the most part, we describe electron flow using the terms oxidation and reduction. When something gets oxidized, it loses electrons, and when it's reduced, it's gaining electrons. Your core metabolism is oxidizing that salad bowl you just ate and reducing the oxygen you breathe. I'm years into writing about biochem, and I still have to remind myself about which word is which. And I use the classic mnemonic here to help me remember. Oil rig. Oxidation is loss, reduction is gain. Oxidation is loss, reduction is gain. Chant that to yourself a few times. Turn it into a meditative mantra. See what new heights of clarity you can achieve. But anyway, the interesting part here is level 2. how are we moving electrons from food metabolism to this big old block of iron compounds? At this moment, it's important to remember that most prokaryotes like GEOBACTER are double layered. You've got the internal cytoplasm surrounded by a barrier membrane, and then that is also covered by another membrane. You might remember some species like mycobacteria take this to a really intense level with their super thick outer mycomembrane. But for the most part, this double membrane allows for a place where the power stroke of metabolism can happen. This space between GEOBACTER's inner and outer membrane is called the periplasm. Down here, beneath the inner membrane, GEOBACTER sulfureducens will burn acetate and generate those electron carriers, which will then gradually shuttle them up to this inner periplasmic membrane. And here we've got some familiar faces. GEOBACTER sulfureducens, honest to God, uses the same complex one you do to shuttle electrons along the cytoplasm. But that's where the similarities end. The real workhorse of GEOBACTER's electron transport chain is going to be this guy, IMCH, which grabs electrons from carriers and also helps shuttle protons into the periplasm so that ATP synthase can use proton flow as its energy source. The wild thing about critters like GEOBACTER here is, depending on the situation, this guy can hot swap different electron transporters into its periplasm. Carriers like CBCl or CBCA are better suited for different electrochemical situations. Keep that idea in your back pocket for a second, we might come back to it later. But here's where things get really wild. This might be hard for critters like you and me to wrap our heads around, because GEOBACTER's respiratory model is basically backwards compared to ours. You and I keep things simple by having lungs and red blood cells that ferry oxygen from the outside of our body to every corner of every tissue. We have the good sense to bring our electron acceptor to the factories that need it. GEOBACTER has to send its electrons. GEOBACTER is going to have to do a lot more work to keep the electron flow going. So the periplasmic space here in GEOBACTER sulfureducens is chock full of these wild, heavy-duty electron carriers, a whole alphabet gang of PPC proteins. These little globs are stuffed to the gills with heme groups to help ferry electrons. You've got electron carriers just like this inside your mitochondria, but as a proud eukaryote, your situation is a little more sleek and refined, using this fella, a nice uncomplicated cytochrome C with a single heme group. And yes, quick aside, this is the same heme group your red blood cells rely on to ferry oxygen. GEOBACTER's PPC carriers look pretty crowded with heme, but if you think this is a lot, then the last few minutes of this video are going to blow your mind. Buckle up, heme fans. The PPC pool here will carry electrons up to the outer membrane of GEOBACTER, where there are, depending on the situation, tons of different potential electron carriers to finish the journey here. GEOBACTER is going to have to put a ton of work to actually bring these electrons home. And this is the most incredible thing about GEOBACTER sulfureducens. In order to connect to its electron sink, GEOBACTER produces one of the most sci-fi-ass biological structures known to science. Behold, bacterial nanowires. Y'all know how electricity works, right? A wire is a piece of metal that connects an electron source to an electron sink, technically. Like, electrons move through the metal and power like a lamp or whatever. We figured out how to pass electricity through wires around about 300 years ago. But critters like GEOBACTER have potentially been working through that part of the tech tree for billions of years. Maybe. You know, that feels like an oversimplification I'll get torched for. Anyway, look at these things. Nanowires are delicate little protein packages encasing a little chain of heme groups. Specifically, a single nanowire subunit is basically a jumbo-sized cytochrome C stuffed with 6 heme groups. Based on the information we have so far, GEOBACTER has a pretty simple philosophy. When in doubt, just throw more heme at the problem. You know, something you can buy anything as long as you're paying the iron price. Anyway, electrons are pushed onto the nanowire by the PPC gang, and then as long as the other end of the nanowire is in contact with a place with positively charged iron 3 ions, that charge difference will be enough to pull electrons along this cute little relay race across each of these heme groups. It is amazing how conductive these little nanowires are. All GEOBACTER has to do is produce a bunch of nanowire subunits like OMCS here and hurl them outside the cell, where the subunits will self-organize like magnetic Legos into these wire shapes thanks to the magic of protein polymerization. This way, GEOBACTER can constantly produce more nanowires and probe deeper and deeper into its, air tank equivalent. But again, how does that work? And so this is the part where GEOBACTER breathes metal, or rather respires metal. Each iron 3 ion that GEOBACTER shoves an electron onto gets converted into iron 2, or rather it gets reduced to iron 2. And wow, neat. That's actually a great way of illustrating why we call this process reduction. We're reducing the charge, making it more negative. Neat. And this is why nanowires make sense. Eventually, this whole pocket of usable iron is going to get reduced to iron 2, which has a less positive charge and therefore doesn't pull on electrons as well. GEOBACTER needs to be constantly building new nanowire networks to keep probing different parts of their local environment. They are constantly depleting their air tank equivalent and need to keep hunting for more electron acceptors. But before we get too far in the weeds, let's pull back and just review at a high level how GEOBACTER pulls off its respiration. So GEOBACTER metabolizes its food, preferably vinegar or the acetate ion, and then shuttles electrons to its inner membrane. Those electrons travel across a bunch of carrier proteins that use the electron flow to pull protons into this periplasmic pocket, thereby generating usable energy for the bacteria. Heme-stuffed electron shuttles like the PPC gang transfer electrons from these carriers up to the outer membrane, passing them onto nanowire networks. Eventually, those electrons arrive at this lovely little iron-3 deposit. Those iron-3 ions are more than delighted to suck up those electrons, and that little pull powers the entire electron relay race. With no iron up here, nothing is pulling the electrons from nanowires, which then can't accept electrons from the PPC gang, which then can't pull electrons from the inner membrane. No electron pull, no ATP power, and that's the single-cell equivalent of suffocating, folks. And congratulations, you have a decent surface-level understanding of the metabolism of GEOBACTER sulfurreducens, one of the wildest current examples of what could have been the original metabolism on Earth, or at least part of the original metabolic picture of ancient life on this planet. And therefore now we finally have enough context to return to this Mars data. Surprise, y'all, this wasn't clickbait after all. Better understanding bacterial respiration helps us make better sense of these rock spots. Now, we've barely scratched the surface of our current understanding of metal-reducing microorganisms, but we have enough to work with, so let's just slam through this thing. Remember, at the simplest level, GEOBACTER sulfurreducens is going to reduce iron-3 to iron-2. That's the key step here. Iron-2 has the perfect charge to join up with phosphate ions, which leads to the creation of crystals like vivianite. So, if the hype bears out here, these little spots are the ancient remains of a dissimilatory metal-reducing microbe gradually exhausting its air tank of electron acceptors. And I promise, one last tangent here. The material in the middle of these spots, gregite, also implies one more step: reducing sulfur ions into sulfide. Iron isn't the only element anaerobes can breathe, sulfur is also a big contributor here. On Earth, we can find evidence of sulfate reducers and iron reducers in close proximity just like this. But I'm not about to do a whole new spiel on a completely different rock folks breathe. Sound off in the comments if you really want me to do a part 2 here covering the second half of this microbial puzzle, or better yet, go to patreon.com/clockworkshow and harass me there. That will really stack the deck in your favor. But yeah, let's go ahead and put a bow on this thing before I get too carried away. I could gush so much more about how powerful this little metabolic trick pulled by GEOBACTER sulfurreducens is. Like, real quick, obviously rocks aren't going to be consistent, so what's our hero going to do when there's different metals with different electric potentials in contact with the nanowires? Well shoot, GEOBACTER will just swap out that IMCH electron transporter for something like CBCl or CBCBA, which is going to be better suited for a less than perfect electric potential. Like, game that out for a second. Imagine if your mitochondria were fast enough to swap out your electron transport chain so you were better suited for breathing something besides oxygen. Also, GEOBACTER sulfurreducens can use those nanowires to move electrons between other community members, making a whole electric network of cooperating individuals as the bacterial colony explores new breathable pockets of metal. But the most important thing here is simply respecting how GEOBACTER demonstrates the core maxim of this channel: life just works. If you can find an electron flux and keep it going, you can build a whole biosphere off that flow. Which is why finding vivianite on Mars is such a big deal, if it actually turns out to be real and also from like a biological source. Organisms like GEOBACTER probably evolved this mode of metabolism on a pre-photosynthesis version of Earth. Our most ancient ancestors took advantage of the incredibly active geology of this good Earth to catch natural electron flows coming out of the cracks. But the Earth is so dynamic and in such constant motion that we simply cannot find rocks old enough to give us concrete answers to some of the foundational questions we have in evolutionary biology. There are tons of great theories that fill in the gaps enough to explain how life could have begun here, but if similar processes allowed a completely separate tree of life to evolve on Mars, then the red planet might just be a critical conduit that will help us understand our own history. That is, if these results hold true. The only way to know for sure is to boldly go, to get enough Martian dirt into a human lab for us to do more robust testing. But like, what an exciting time to be in this business, y'all. That's a good place to leave this here, though. I really appreciate your time. Thank you so much for being here. And hey, thank you so much for reaching the end of this video here. If you like this video, please make sure you like, subscribe, and leave a comment, as that's the thing that's going to really help us stand out in the new YouTube meta. More importantly, it would mean everything to me if you went over to patreon.com/clockworkshow and checked out all the cool stuff we're building over there. Heme fans are going to absolutely love the sticker pack we have for October, and the molecular paper craft I am producing for this month is also really special. Check us out there. I can't wait to show you guys what I've been working on, and I can't wait for you to be a part of the amazing community we're building over there. At the same time, you can always check us out over at watchclockwork.com and join our email newsletter, which will eventually achieve consistent production. I promise, it is happening. Otherwise, I don't want to keep you too long here. I really appreciate your time. Thank you so much for being here, and as always, I like to leave you with peace, love, and heme without end. Everyone be well. Thank you so much.